Hello everyone, myself Dr. Girish Adi, working as Assistant Professor in Department of Plant Pathology, School of Agriculture, SR University. Today we'll discuss about diseases of maize and their management. So coming to maize plant, it is an important cereal crop in the world. Coming to important diseases in maize, first we'll start with the downy mildews in maize. There are different types of downy mildews reported in the maize. We will see one by one. First is downy mildew caused by Pernosclerospora sorghum. There are different strains of Pernosclerospora reported in maize. One is Thai variant or sorghum downy mildew, Pernosclerospora sorghum. Then Java downy mildew, which is caused by Pernosclerospora Maydis. Then Philippines downy mildew, which is caused by Pernosclerospora philippinensis. Rajasthan downy mildew, which is caused by Pernosclerospora heteropogoni. Then sorghum downy mildew, Pernosclerospora sorghum. So coming to characteristic symptom of downy mildew in maize, the most characteristic symptom is proliferation of leafy structure from the ears and tassels. In many cases, the leaf protrusions occurs only the ears, resulting in the mass of strap-like leaves protruding from the ear zone. Affected plants may also have profuse tiller development. So this all we can call it as a crazy top symptom, complete proliferation of the leafy structure from the ears or tassel. So coming to other symptoms of downy mildew in maize, the first, this has a very wide O strain. The first symptom to appear is chlorosis of the leaves. As the plant grow, it often shows stunting and necrotic lesion begin from the leaves. As the leaves continue to grow, wide downy growth may develop in the necrotic lesion, especially on the underside of the leaves. The downy growth is result of production of conidia and conidiospores under the proper environmental condition. Sorghum and maize express very similar symptom. In sorghum, chlorosis of the seedling is very common after infection. As the leaves get older, they can express white striping, which eventually leads to necrosis of the white striped tissue. When the leaves die, they begin to become shredded in the appearance, similar to hail damage. The symptom is associated with the production of oospore in the leaf tissue. If the sorghum plant are young and produce conidia on their leaves, this type of infection often leads to the death of the entire plant. You can see the most of the symptom in sorghum and maize are similar. Coming to the key factor in diagnosing the downy mildew on maize and sorghum caused by Pernosclerospora sorghum include the chlorosis of the leaves, followed by white streaking of the leaves. The leaves could also have white downy growth underside of the leaf, and the white streaked areas of the leaves become necrotic over time, leading to leaf shedding symptom. Leaf shedding is the most characteristic symptom, and it is the final stage of the downy mildew. In maturing plant, the reproductive structure do not form properly or do not form at all. This leads to sterile plant, and also sometimes crazy top development also seen on downy mildew in maize. So in this picture we can see formation of the oospore on the shredded leaf. These oospores are the sexual resting spore of the fungus, which are survive in the soil over a period of time, and these will serve as the primary source of infection. This one is the disease cycle of maize. Coming to maize, downy mildew disease cycle. Pernosclerospora sorghum is a polycyclic disease cycle. It is capable of causing secondary infection of susceptible host throughout the growing season. Its resting structure, the structures that allow the pathogen to overwinter, are the oospore. These oospore produced in the infected plant from the previous growing season. They are often disseminated by the wind. The oospore can overwinter in the soil, in the debris, and the surface of the soil. The oospore have very thick wall, which makes them capable of surviving in the soil for years together under many different weather conditions. It is also possible for oospore and mycelium to overwinter in the seeds of the maize. The mycelium infect the scutellum of the seed. The oospore and the mycelium that are present in the seed often lose their viability when the seeds are dried, but under the right circumstances, it is possible for these infected seeds to become a source of inoculum. Infecting the maize plant as it grows. Infection of the seed itself often occurs with the plant that produced and the seed had been infected later in the development. The oospore are main source of primary inoculum in the disease. They present in the soil when the host seedling are germinating. The oospores then infect the roots of the seedling. This type of infection is a systemic infection of the plant. The pathogen grows throughout the plant, infecting the leaves as they grow, leading to chlorosis. The chlorotic leaf develop a white streak. These white streaks are the location of oospore production. This only occurs in the plant that were systemically infected at the seedling. When the oospore become mature, the white streaks on the leaves turn to brown and become necrotic. These necrotic areas become shredded over time, which is how the mature oospore are disseminated. The spores are carried by the wind and they become source of inoculum in the subsequent generation. As the pathogen continues to develop in the host plant, they may also lead to production of conidia on the leaf surface. It is the conidia or conidia 4, it causes the white downy growth. We can say sporangia or sporangiospore or conidia 4 also. After they are released, they are wind disseminated, and these sporangiopore and sporangia, they will cause secondary infection. So this is the whole disease cycle of downy mildew of maize. So here the main primary source of inoculum is oospore present in the soil, overwintering oospore, and the secondary spreading mainly through zoospore. Secondary zoospore, they may spread through irrigation water or rain splash, they will cause secondary infection. Then coming to management of downy mildew in maize, seed treatment with metalaxyl fungicide at the rate of 6 gram per kg or Epron 35 WP at the rate of 2.5 gram kg. Rogue out the infected plant at the early stage and spray with the metalaxyl 1 gram per liter or metalaxyl plus mancozeb at the rate of 2.5 gram per liter. It is the most effective management. Apart from the chemical management, you can also go for crop rotation with a non-host crop like rice or any other pulse crop, and also go for clean cultivation and deep summer plowing to expose hidden oospore to the solar radiation. And also we can practice other biocontrol agent like application of trichoderma arsenum and pseudomonofluorescence at the time of sowing. So these are the IDM practice recommended for the control of downy mildew disease in maize. So coming to other types of downy mildew, one is Philippines downy mildew, which is caused by Pernosclerospora philippinensis. Then Java downy mildew, which is caused by Pernosclerospora maydis. Then sorghum downy mildew, which is caused by Pernosclerospora sorghum. We already studied about this disease. Then sugarcane downy mildew, this is caused by Pernosclerospora sakari. Coming to another type of downy mildew, which is brown striped downy mildew, which is caused by Scleroptera rhizia or rhizia. Coming to the major symptom, lesions start developing on the lower leaves as the narrow chlorosis or yellow stripes of 3 to 7 mm wide with a well-defined margin and are delimited by veins of the leaves. The stripes later become reddish to purple. Later development of the lesion causes severe striping and blotching. Seeds development may be suppressed. Plant may die prematurely if blanching occurs prior to flowering. Sporangia on the leaves appear as the downy whitish or woolly growth on both the surface of the lesion. Floral or vegetative parts are not malformed and leaves do not shed. So this is the completely different type of symptom when compared to sorghum downy mildew in maize. So this is the picture representing brown striped downy mildew. Coming to management, here the resistant varieties like Prabhat, Kohinoor, ICL, PAC 9401, PMZ2 and CTEC 2331 are the available resistant varieties. Then we can also go for seed treatment with the metalaxyl at the rate of 6 gram per kg. Rogue out the infected plant at the early stage of infection. Can control with Epron 35 WP at the rate of 2.5 gram per kg as a seed treatment. Spray with the metalaxyl or metalaxyl plus mancozeb at the rate of 2.5 gram per liter of water. So this is regarding integrated management of brown striped downy mildew in maize. So coming to the next disease, charcoal stalk rot in maize, which is caused by Macrophomina fasciolina. This charcoal root rot or charcoal stalk rot, this is known as dry root rot. Why? Because this disease or this pathogen favors by high temperature and dry period. So the temperature in the range of 35 degrees Celsius followed by dry hot period favors the disease infection. Coming to major symptoms of charcoal stalk rot, disease appears 1 to 2 weeks after flowering, which is also known as post flowering stalk rot. The outside of the lower internode becomes dark colored, the pith becomes badly disintegrated. The pathogen invades the seedling root. When the plant approaches maturity, the internal parts of the stem show black discoloration and shedding of the vascular bundles. So this occurs mainly in the lower stalk internodes. Careful examination of the ring and vascular bundles of the infected plant reveals a small black sclerotia which can overwinter and infect the next crop. Fungus may infect the kernels which cause them blacken completely. Disease is favored by high soil temperature of 30 to 42 degrees Celsius and low soil moisture. So low soil moisture combined with a high temperature and not and like without any proper nutrition to the crop always favors the charcoal stalk rot. So these are the images representing the charcoal stalk rot, complete disintegration of the vascular tissue, complete turning of black color appearance of the vascular bundles. Sometimes the pathogen may also infect the developing kernels also. So this is the microsclerotia which is able to survive in the soil for longer duration. So this is the disease cycle of charcoal stalk rot. First the microsclerotia which are present in the soil which will cause the primary act as a primary source of inoculum. This germination of microsclerotia at the temperature of 30 to 35 degrees Celsius, they will germinate and they will cause infection to the root system, and germ tube formation will occur and development of aprosuria, then penetration of the word system, then invasion of the tissue. Hyphae and microsclerotia disrupt the water and nutrient transport to the upper parts of the plant, that will completely block the movement of water and nutrient, and the plant will exhibit the typical symptom in the plant like yellowing and running of the leaves that remain attached to the stem by petiole, grey appearance of the lower stem and taproot due to abundance microsclerotia. Then premature death of the plant will occur and survival of the microsclerotia in the root system and the plant debris will occur. Then again the pathogen will continue the life cycle. Coming to management, maintaining the soil moisture during the first flowering period to minimize the stalk rot, balanced application of the NP and K fertilizer, avoid high plant population, grow drought tolerant, lodging resistant, non-senescence sorghum cultivar, soil application of carbendazim at the rate of 2 gram per liter, apply trichoderma in a furrows after mixing with FIM at the rate of 2.5 kg per hectare, or we can also go for application of trichoderma along with the oil cakes like neem cake, pongemaya cake or any other manures. So coming to next major disease in maize, which is banded leaf and sheath blight in maize. So coming to this banded leaf and sheath blight, which is caused by Rhizoctonia solani. So coming to important symptom, the most characteristic symptom include the concentric band rings on the infected leaves and the sheath that are discolored, brown, tan and grey in color. Typically, disease develops on the first and second leaf sheath above the ground and eventually spread to ear, causing ear rot. Ear rot is characterized by the light brown cottony mycelium on the ear and the presence of small round sclerotia which are nothing but a compact mass of hyphae that can survive in unfavorable condition. Ear will dry prematurely and caking of the ear sheath is common symptom. So these are the images representing banded leaf and sheath blight. You can see the lower leaf sheath is get infected by the fungus, formation of a grayish like straw colored irregular margin appearance on the lower leaf sheath. Then eventually the pathogen will cause infection on the cob or ear. So this is the culture plate representing the culture of Rhizoctonia solani. Most identifying character of this pathogen is right angle branching and formation of macrosclerotia. So these are all the symptom development photos. Initially there is a straw color with a small spot formation will occur. Eventually it will cause complete blighted appearance or complete burnt appearance to the crop. So coming to the disease cycle of Rhizoctonia solani, first the majorly the mycelium which is present in the the macrosclerotia which are present in the soil, they will come in contact with the lower leaf sheath. So this lower leaf sheath get infected and there is a complete infection to the lower leaf sheath and development of the symptoms occurs. Then after that ear infection will occur. Rhizoctonia solani overwinters as a spores in the soil. Spores is nothing but a macrosclerotia. After that, the inoculum present in the soil which is coming in contact with the young seedling and it will cause complete symptoms like banded leaf and sheath blight. Coming to management of banded leaf and sheath blight, avoiding water stress at the flowering time to reduce the disease incidence. Apply trichoderma in furrows after mixing with the FIM 2.5 kg per hectare. or we can also go for application of trichoderma along with the vermicompost or any type of oil cake like pongyama cake or neem cake. We need to mix 10 days before sowing in the field condition. So this is regarding banded leaf and sheath blight. Then we will see about common smut of maize which is caused by Ustilago Maydis. Coming to symptoms, gall develops on the ears and leaves and stalk and tassel. These galls initially covered with a thin white silvery membrane. Interior of the galls develop into dark mass of dark spore which is nothing but it contains teleospore. This pathogen is present where the corn is grown around the world. It can occur throughout the season. So these are the different types of symptoms of the common smut in maize. On the smut of sweet corn, you can see formation of the galls on the infected corn. These galls initially covered with a thin white silvery membrane. When this membrane ruptures, it will release the black powdery mass of spore. These spores nothing but a teleospores of the pathogen. There are different smut types of smut present in the maize. Coming to its smut, which is caused by Specialotheca reliana. Coming to symptoms, ears and tassel may be replaced with a smut spore filled with the teleospore. Infected plant may develop Bezier symptom on either ear or tassel, including the production of wire-like extension of vascular bundle in the tassel or finger-like proliferation in the ear or cob. Ears of the infected plant may be recognized by teardrop shape and lack of silk where the husks are filled with the teleospore, which is a major symptom of head smut. So these head smut spore is nothing but teleospore. We can see electron microscope image of single cell teleospore. Then coming to disease cycle of maize smut. So common smut, the disease occurs whenever the corn is grown caused by Ustilago Maydis. The fungus survives mainly on the crop debris and also in the soil. It can infect any type of issue plant by entering through wounds and forming characteristic smut galls. Mainly this fungus Ustilago Maydis disease cycle follows a disease cycle where the teleospore overwinter in the soil or crop residue, spreading via wind or rain that can infect any young plant tissue during growing season. The fungus creates a tumors or galls and the galls which mature into black powdery mass of spore and releasing the repeating cycle. So after releasing the teleospore, it will repeat the cycle and it continues the life cycle. So coming to management of smut of maize, crop rotation of the pulses, treat the seeds with Captain or Thyram, 4 gram per kg. Planting date may be also altered in some time areas like early sowing, which can control the infection with the smut fungus. Late sown crop is usually affected by the smut disease. Then coming to rust disease in maize, this common rust in maize which is caused by Puccinia sorghum. Coming to major symptom, pustules are which are oval to circular and red to dark brown in color. Pustules are common on both the leaf surface. Rust spore like iridospores are cinnamon brown in color. Leaves as well as sheaths can be infected under severe condition and leaf chlorosis or death may occur. Coming to pathogen, which mainly overwinters in the southern US or Mexico, not in the corn belt. Iridospores serve as a repeating spore. Commonly Puccinia will produce 5 types of spore. One is pycnospore, Aciospore, Iridospore, Teleospore, and Basidiospore. So among these, Iridospore, which will cause repeating infection in the life cycle of the crop, we can say this Iridospore is a repeating spore. So these are the pictures representing formation of rust pustules on the mainly on leaf sheath. So this is the disease cycles. Majorly, first the iridospores present on the environment, they will cause infection to the maize plant. So these iridospores we can call it as repeating spore will cause repeated infection to the crop in a life cycle. Iridospores are wind blown to corn on the spring in summer months. Infected leaves produces a iridospore and it will continue the repeating infection. When the plant reaches to maturity, the pustule mature, there is a formation of teleospores will occur. These teleospore overwinter and germinate in the spring season, produce basidiospore, which are spreaded by the wind which are carried by the wind, and they can infect the alternate host Oxalis corniculata. So these Oxalis corniculata get infected by the basidiospores. So when basidiospores, after that these Oxalis corniculata will produce the sexual spores like pycnospore and achiospore. So these achiospore they become windborne and which can infect the new maize plant. So like this it will complete the life cycle. So here the major host is maize plant and the secondary alternate host is Oxalis species. So this is how the pathogen Puccinia will complete the life cycle in maize rust. So coming to management, mainly we can follow chemical management like application of mancozeb 1.25 kg per hectare. Or else we can also go for triazole fungicide like hexaconazole, propiconazole, topiconazole at the rate of 1 ml per liter. And other management practice like crop rotation and clean cultivation, deep summer plowing, all are recommended to control the common rust disease in maize. So coming to another important foliar disease, Maydis leaf blight in maize. So this Maydis leaf blight in maize, which is caused by bipolaris maydis or Coccylobulus heterostropus. Coming to the major symptom, young lesions which are small and diamond shaped. As they mature, they elongate. The lesion may coalesce, produce a complete burning of the large areas of the leaf. They vary in size and shape among the inbreeds and hybrids with a different genetic background. The race O of this fungus produce tan elongated lesion between the vines with limited margin with a buff to brown border usually attacks only leaves. So this is the picture representing the Maydis leaf blight caused by bipolaris or Coccylobulus. So these are the different picture. Infection may also seen on leaf sheath and also cap. So we can see the clear cut diamond shaped spots appears on the leaves. Later on it will give complete burnt or blighted appearance. So this is the spore produced by Coccylobulus variza. This is the severely infected flower with a Maydis leaf blight. Coming to management, mainly chemical management like application of mancozeb 2 gram per liter and some resistant varieties like Deccan, VL42, Prabhat, KH5901, Pro 324 and Paraz, Sartaj, Deccan 109 which are act as a resistant varieties. So coming to another important foliar disease, Turcicum leaf blight in maize. This Turcicum leaf blight it is caused by Exeroleum tursicum, which is also known as northern corn leaf blight. So coming to major symptom of tursicum leaf blight, it causes significant economic damage and the infection takes place at silking stage and conditions are optimum. In the early stage, slightly oval water soaked small spot produced on the leaves. These grow into elongated spindle shaped necrotic lesion. They first appear on the lower leaves and continue to increase in size and length as the number of the plant develops until it gives a complete burning effect of the foliage. The spots which are very long, elliptical, grayish green, tan lesion ranging from 2.5 to 15 centimeter in the length. So these spots we can see very long elliptical lesion with a 2.5 to 15 centimeter in length. And these are the spores of Exeroilium tursicum. Initially the spots are water soaked very less in size. Later on they will grow and they will go on increasing their size. So coming to management of Turcicum leaf blight, we can apply chemical fungicide like mancozeb, carbendazim or any triazole fungicide like hexaconazole, propiconazole at the rate of 1 ml per liter. Some resistant varieties like Deccan 105 and Deccan 109 and JH 1267 are recommended. Coming to important other important disease in maize like post flowering stalk rot. Like first one is Pythium stalk rot, which is caused by Pythium apanodermatum. Coming to the major symptom, usually the basal internode becomes soft, dark brown, water soaked, causing the plant to large. Damaged internodes commonly twist before the plant large. A diseased plant can remain alive until the vascular bundles become affected. Isolation in culture media are necessary to differentiate the Pythium from Erwinia stalk rot. So come to management, planting time between 10 to 20 July in northern India, maintain plant population around 50,000 per hectare. Um, Good field drainage, removal of previous crop debris, soil drains with a captain at the basal internodes, or we can also go for carbendazim or mancozeb. And also best effective fungicide is metalaxyl along with the mancozeb. Resistant varieties are like Ganga and Sulphate 2. Coming to another important stalk rot, Fusarium stalk rot caused by Fusarium moniliforme. So coming to major symptom, affected plant wilt and leaves change from light to dull green and lower stalk becomes straw colored. Reddish discoloration occurs inside the infected stalk. The internal pith tissue disintegrated, leaving only vascular bundles. Fungus enter through roots and grow up in lower stem. If the infection occurs just after flowering, husk appear bleached and straw colored. Coming to management, application of potassic fertilizer reduces the infection. Seeds from infected area should not be planted. Rotation with other crops we can follow like other cereal crop. Resistant varieties like Ranjit and Ganga fire recommended for cultivation. So coming to other disease in maize like anthracnose, leaf blight, which is caused by Glomerella graminicola or Coltotrichum graminicola, which is a Glomerella is a sexual stage, Coltotrichum is a asexual stage of the fungus. Coming to symptom, small oval to elongated water-soaked legend enlarged to become brown spindle-shaped spots with a yellow reddish-brown border. Legend may coalesce and blight the entire leaves will occur. Other legend will turn grey in the center with a small black specks. These small black specks which are representing the acerulae with a sterile black ear. These acerulae are nothing but asexual fruiting structures of the fungus. Leaf blight may be followed by top kill and stalk rot. Leaf blight rarely causes large yield losses. Stalk rot phase is most important in anthracnose. So these are the images representing anthracnose leaf blight on the lower leaves early season. Anthracnose on the leaf blight, it will cause leaf infection. Then anthracnose like stalk rot symptom. So these are the stalk rot symptom caused by the fungus Coltotrichum. So coming to favorable condition, favored by cool to warm weather, wet humid weather, continuous corn with reduced tillage. Inoculum survival, mainly it survives on the infected plant debris like leaves, leaf sheath, stalk or seed endosperm. Inoculum dispersion mainly through airborne spore. Coming to management, resistant hybrids, application of chemical fungicide like carbendazim, hexaconazole, propiconazole at the rate of 1 ml or 1 gram per liter. Cleanly plow under infected residue and deep summer plowing. So all these IDM practice we can recommend to control the anthracnose or leaf blight disease in. Coming to next important disease, bacterial stalk rot, which is caused by Erwinia chrysanthemi pethavorsia. Coming to the symptoms, the stalk near the ground become water soaked with a brownish discoloration and are easily breakable. The rotting of the tissue, which will emit a very putrid smell of the bacteria. Infected plants show dark color and water soaking at the basal end of the stalk. The plants die shortly after tasseling. The bacterial decomposition produces an unpleasant odor, like a putrid smell of the bacteria. So coming to maize management, like bleaching powder containing 33% chlorine, we can apply at the rate of 10 kg per hectare as a soil drench or flea flowering stage. Planting crop on the ridges, avoid the water logging, giving proper drainage, which can effectively manage bacterial stalk rot. So coming to important viral diseases in maize, first one is maize dwarf mosaic. Coming to the major symptom of maize dwarf mosaic, the symptoms often begin as a chlorotic spot and the streaks on green young leaves, which later develop into mottle or mosaic pattern. The viral strain, the corn genotype, and the stage of the corn development at the time of infection will affect the different types of symptom. Upper internode of the corn may be shortened and excessive tillering may occur. Ear formation or development may very slow, which may cause grain yield loss. Hybrids infected early in their growth stage may be stunted. So these are the, and also we can see a formation of like chlorotic patches with a mosaic pattern on the infected leaves. So completely the plant look very stunted in growth and sometimes there is no reproductive growth. So all these are the important symptom of maize dwarf mosaic. So you can see the stunted growth of the plant along with the chlorotic patches and the mosaic pattern. Coming to the strains of maize dwarf mosaic, strain A, the major source is Johnson grass, and the strain B, here the Johnson grass is not a host. Main transmission will occur through apid species, and corn leaf apid is a primary vector. So mostly these apids will transmit the maize dwarf mosaic virus in a non-persistent manner. Here, in a short period of time, vector will acquire the virus and it will transmit it in a very less time, and there is no multiplication of the viral particle inside the vector. So this type of transmission, we can say it is non-persistent manner of transmission. So coming to maize dwarf mosaic etiology, it is a party virus. This pathogen consists of 4 strains C, D, and F. Majorly aphids can transmit the maize dwarf mosaic virus in non-persistent manner. Can be C transmitted at very low frequency or mechanically transmitted by leaf rubbing, etc. Will can transmit the disease, which will majorly infect Johnson grass and also sorghum. So these Johnson grass and sorghum, they will act as a the other like non-host crop, which will also the pathogen will cause infection. Then coming to time of occurrence, symptom appear in the 6 weeks after apid feed on and transmit this virus. Condition favoring the disease, average to warm temperature, which is about within 28 to 30 degree, and nearby Johnson grass infected with the maize dwarf mosaic virus may increase the disease. Mainly this Johnson grass and sorghum, they will act as a collateral host for the maize dwarf mosaic virus, because this all these Johnson grass, maize and sorghum, they will belong to same family. Coming to maize streak virus, this maize streak virus, which is the first to reported in the East Africa in early 1900 and remains a major constraint in the maize production in Africa. This maize streak virus transmitted by several species of Cicadolina leaf hoppers. So here the leaf hoppers, which will act as a major vector. Coming to the symptom, disease symptom first become apparent about a week following infection, and a small chlorotic circular spot arises on the basal section of the young leaves that emerge after infection. Spot contrasts sharply with surrounding healthy green tissue. Number of spots increase with the plant growth and can coalesce. In susceptible varieties, spots enlarge parallel to the leaf lines, forming a distinctive elongated chlorotic streaks and distributed evenly over the leaf surface. Infection with the maize streak virus commonly result in crop stunting and barren plants without any reproductive growth. So these are the symptom of maize streak virus. We can clearly see formation of chlorotic streaks on the infected leaves and chlorosis on the affected plant. Coming to host major IDM practices, first one is host resistance. Cultivation of the resistant varieties offer a most practical and cost effective management practice. Resistant varieties are widely available. Resistant varieties have been developed by ITA and CIMIT, adapted for a wide range of growing conditions in Africa and also India. Coming to vector management, the management of vector population through insecticide, like systemic insecticide, we can like imidacloprid and imidacloprid or fipronil or any other systemic insecticide we can recommend to control vectors like aphids and leafhoppers. Coming to cultural methods, this maize streak virus is known to infect a wide range of gramineous species, and there are few cultural practices that can reduce the disease severity. Mainly, we can go for crop rotation with other pulse crop or other non-host crop to control maize streak virus. So this is regarding important diseases in maize, like starting with the downy mildews in maize, maize smut, then maize rust, and important post flowering stalk rot in like Erwinia stalk rot, Pythium stalk rot, Fusarium stalk rot, and along with the viral diseases like maize dwarf mosaic and maize streak virus and their integrated management. Thank you.